Okay, the recording is now on. Welcome to our new series called Charging Forward Virtual Medical Symposium Series. We created this program because we want to give you the opportunity to hear the best information on VEDS or Vascular EDS from our professional advisory board and other leading medical experts. We are grateful to the Chu and Chan Foundation for making these webinars possible. If you have not already visited our website, we hope that you check it out at thevedsmovement.org and find the resources helpful. Before we get to our presentation and our introduction of Dr. Dietz, I'd like to introduce Michael Weimer, the President and CEO of the Marfan Foundation, to welcome you.
Thanks, Katie. Good evening, everybody. I actually wanted to combine, well, I actually wanted to hear Hal this evening, but I also wanted to combine and say hello and wish everybody good days and healthy days as we roll forward here. As I mentioned, if you were on a few minutes ago, I think of Dr. Dietz as America's favorite webinar host. So you're in for a real treat this evening. I'm also really excited about what has been accomplished with our VEDS division. I think it actually started, we often try to figure out the exact date, but I think we can all say the beginning of October is when we began our efforts. And I think what's happened between October and now has really been fantastic. And, that's been a combination of Katie as the director of the division, Joe Grima, who is our chief science officer, who is remarkable in everything she does. And we also have a great steering committee. We have the support and confidence of somebody like Kathy Bowen, who is really at the very core of the whole VEDS movement. So I'm very appreciative for what we've done and for the people that are involved. But, the battle's just beginning. And in fact, the biggest part of our journey is still way out ahead of us. And we need to do more and we need to find answers. And tonight, with Dr. Dietz here, I ultimately believe that the answers will come from people like Hal. And those of us that are engaged in this day in and day out. So again, just wanted to say hello. I'm going to be here for the evening and stay healthy. And Katie, back to you.
Thank you so much, Michael. I know that the community and certainly myself very much appreciate the Marfan Foundation and the VEDS movement. So thank you very much for welcoming us. As you know, tonight's presentation is on genetics and VEDS. If you have questions during the presentation, please type them in the Q&A box, which is different than the chat box. You can see the Q&A box on your screen, and we will leave time at the end to answer questions. So I'm very excited now to introduce our speaker tonight, Dr. Hal Dietz. Dr. Dietz is the Victor A. McCusick Professor of Pediatrics at the Department of Genetic Medicine at Johns Hopkins University School of Medicine and an investigator at the Howard Hughes Medical Institute. He heads a multidisciplinary clinic for the diagnosis and management of individuals with heritable forms of cardiovascular disease, with a special emphasis on VEDS, Marfan syndrome, and related connective tissue conditions. He directs a group of dedicated molecular biologists focused on improvement of the lives of individuals with these conditions through the development of new diagnostic and treatment strategies. A member of our professional advisory board, Dr. Dietz focuses his research on explaining the causes and pathogenesis of connective tissue conditions that involve the cardiovascular system. His lab is best known for in-depth studies that involve the creation and characterization of animal models. Now we're ready to start the webinar, and I will pass control over to Dr. Dietz. Thank you so much, Dr. Dietz.
Thank you, Katie, for that really generous introduction. You cannot start sharing screen while other participant is sharing.
Okay, try it now. There we go.
Can everyone see the first slide with lots of words on it?
Yes.
Okay, so good afternoon or evening, everyone. It's a pleasure to be with you. I hope you are all well and safe. And it's my pleasure tonight to talk about the genetics of vascular Ehlers-Danlos syndrome. And when I was trying to decide what to talk about in this webinar, I thought it would be a good idea to try to address all the information that someone hears when they first learn about the diagnosis of vascular EDS, when they first learn about genetic testing, when they hear about the implications of genetic test results, and when they talk to other families and kind of compare notes about what they've heard and what the genetic findings may mean. So this is a basic outline of what I hope to cover. First, a very brief clinical description of vascular EDS, a primer in genetics. So I'm going to take you back to high school, and we're going to review some of the fundamentals of genetics that will impact our understanding of a genetic test result. We'll talk about inheritance patterns for vascular EDS, what the type of mutation means and how that can help us interpret what the disease mechanism might be. When we talk about a disease mechanism, that means the sequence of events between someone inheriting a gene change and then someone showing a consequence of that gene change, like a vascular problem. So what are the intermediate steps that are necessary to drive that process forward? We believe if we understand those intermediate steps, then we can try to devise strategies to block them. So that gets to the why this matters section. We'll talk a little bit about why genetic testing might be useful for a given individual or family. Talk about some recent research results that are showing promise. And then finally end up with a brief discussion about other conditions to be considered if someone is thought clinically to have features of vascular EDS, but the genetic testing comes back negative. So as many of you know, vascular Ehlers-Danlos syndrome is an inherited condition. It's caused by a change in the gene in our body that encodes a protein that's called type 3 collagen. The proteins are what are the business end of getting things done in the cell. The genes simply provide information regarding making proteins, but proteins are actually the molecules in the body that do the work. So in vascular EDS, a change in this gene has many predictable consequences, including an enlargement or aneurysm of really any medium to large artery throughout the body. Unlike Marfan syndrome and some other conditions, the base of the aorta, the largest blood vessel carrying blood away from the heart, is typically spared in vascular EDS. So if a family has many family members with aortic root problems, probably another diagnosis should be considered. Now, in vascular Ehlers-Danlos syndrome, an artery can tear or burst, and that this can occur without prior enlargement. There's also risk of rupture of the large intestine and also of the uterus, specifically when a woman is pregnant. There's also an increased risk of operative complications, and this helps to distinguish the management plan for vascular EDS versus that for other connective tissue disorders. There are some more rare but serious medical problems associated with vascular EDS that are quite specific to this diagnosis, something called, for example, a carotid cavernous sinus fistula, which abruptly increases the pressure in the venous system behind the eye and can cause a sudden bulging red and painful eye with blurry vision. This is something for people with this condition to be aware of because it does require quick medical attention. Now let's get into the basics of genetics. So everyone's body has a lot of cells, which are the little factories in our body that actually perform body functions. A cell, based upon its identity, will do different things, but we have about 30 trillion of these cells in our body, and every one of them has 23 pairs of chromosomes, one pair that we inherit from our mother, and the other complement, the other set of 23 chromosomes that we inherit from our father. Each chromosome has 3 billion DNA building blocks. So in total, with 23 pairs of chromosomes, we each have about 6 billion DNA building blocks that we call nucleotides. And the important function of these DNA building blocks is to encode units of information that we call a gene. Now, in general terms, for the purpose of this talk, we can define a gene as a segment of DNA that encodes a specific protein. And we have about, I'm sorry, this is incorrect. It should be 20,000 genes in total, and that's 20,000 pairs. So one that we inherited from our mother and the other from our father. Only one of these genes encodes type 3 collagen, and we call that the COL3A1 gene. We get, again, one copy from mom and one copy from dad. And all it takes is one DNA change, or something that we call a mutation, to cause vascular EDS. So if you can imagine, among the 6 billion DNA building blocks that we have, all you have to do is alter one of them to develop vascular Ehlers-Danlos syndrome. Now, how does a gene tell a cell what to do? This is going to be a little bit complicated, but I'm going to try to simplify it as much as possible, and I'll also try to reduce it to the important information for interpreting a DNA change. But a gene makes a copy of itself that we call RNA. When that copy of RNA is first made, it's immature. We call it a pre-mRNA because it requires processing. You can see these colored boxes. Each of those boxes represents a block of DNA that's contributing to the information regarding how to make the type 3 collagen protein. But you can see that those blocks are separated from each other. The first thing that has to happen to a pre-mRNA is it has to be so-called spliced. That is, those coding building blocks that are colored have to be joined together, and you need to get rid of that intervening sequence of DNA that can be considered junk. So at the end of this splicing process, we have what we call a mature messenger RNA or mature mRNA. So then what happens is a structure that's called a ribosome attaches to the RNA. That's the little blue snowman that you can see found its start point. Its start point is always encoded by the 3-letter code AUG. And then what a ribosome does is it simply reads down the messenger RNA, translating its code into a protein. And in this circumstance, it's going to be the type 3 collagen protein. You can see that each of those colored building blocks of mRNA contributed to a certain region of the protein that's shown by the corresponding color. And you can also see that the ribosome knew where to stop making protein. That 3-letter symbol TER stands for a termination signal, so that it's telling the ribosome its job is done. Now, as I mentioned, we all have 23 pairs of chromosomes. Here, this is a so-called karyotype that it is showing the structure of our chromosomes under the microscope. You can see that the type 3 collagen gene is on chromosome 2. We have one pair from mom, one copy from dad. And you can see that only one of those copies has a change marked by the red X. So when a condition can be caused by a single abnormal copy of a gene, we refer to the inheritance as so-called autosomal dominant. Here I'm showing you little families. A round symbol represents a woman. A square symbol represents a man. An open or white symbol represents someone that's not affected. And a darkened or black symbol represents someone that has vascular Ehlers-Danlos syndrome. And you can see in the family all the way on the left that a father who's 80 years old has passed this on to his son who's 55 years old. And then that gentleman passed on the mutation and therefore vascular Ehlers-Danlos syndrome to his 30-year-old daughter. And the important point is that it only takes one abnormal copy of the gene from one parent to have the condition. On average, for any given child of an affected individual, there would be a 50-50 chance if they were going to inherit the mutation from their parent, because the parent has two copies to give, one that's abnormal, one that's normal, and it's completely random as to whether one or the other is passed on. Now, in the middle family, we have a little bit of a variation on this theme. We have some people in the family who have the mutation, but have mild disease, mild forms of vascular EDS shown by the gray symbols. And then you can see once again that the woman at the bottom of the family, the 30-year-old woman, has classic vascular Ehlers-Danlos syndrome shown by a black symbol. Finally, all the way on the right, we can have a situation again with autosomal dominant inheritance. The mutation is being passed down through each generation. But in the middle, that individual with a dot in the center has no feature of the disease. They have the mutation. They inherited that from their affected father. They passed on that mutation and their daughter has the condition, but that intermediate individual never showed any manifestation of the condition. This is called so-called incomplete penetrance, and we'll get back to possible reasons why this might occur. Now, often you see an individual with a condition, in this example, vascular EDS, who's the first person in their family to have the condition. That's called sporadic vascular Ehlers-Danlos syndrome. And that's typically most often caused by a new mutation. That means during the formation of the specific egg cell or the specific sperm cell that went into that person's conception, a DNA mistake or change was made in the type 3 collagen gene. So that individual would be the very first person in their family to have the DNA change, and also the first person in their family to have vascular Ehlers-Danlos syndrome. Occasionally, you see the exact same thing. Someone that has no feature of vascular Ehlers-Danlos syndrome, no family member has ever been affected, but suddenly there is a child with vascular EDS. And if you do very careful DNA analysis, you learn that the parent, the individual shown with those little tiny black boxes, is a so-called mosaic. That means that a few cells in their body have the mutation. It's not present in every cell of their body. So they don't have vascular Ehlers-Danlos syndrome, but they are at risk to pass that mutation on. In the first example, when you have a new mutation occurring, the risk of having another affected child is exceedingly low, as close to zero as you can imagine. In the mosaic example, the risk is somewhere between very, very low and 50%. It's hard to say without knowing how many cells in that person's body has the DNA change. But a clue to this mosaicism circumstance would be apparently unaffected parents having more than one affected child with vascular EDS. If you just play the averages, if you see a family with the first individual with vascular EDS, about 75% have a new mutation. In about 25% of the families, there is some degree of mosaicism. Another important point in either circumstance, once a child is affected, the risk to their children is now 50%. It doesn't matter if it was a new mutation. It doesn't matter if it was due to mosaicism. The risk moving forward to each child would be 50%. All right, so what are some common misconceptions about the inheritance of vascular Ehlers-Danlos syndrome? We often hear about individuals who've been told that they have a combination of Ehlers-Danlos syndrome types, some vascular with some degree of hypermobile, and some classical, perhaps with a touch of Marfan syndrome. Can this happen? The answer really reliably is no. These are discrete disorders. They're caused by changes in different genes. So therefore, it's highly unlikely that someone has multiple connective tissue diagnoses. It's more likely that a single connective tissue diagnosis will be able to explain that individual. Can the type of EDS jump back and forth between generations in the same family? Someone has hypermobile EDS, they inherit that same DNA change from their parent with hypermobile EDS, and suddenly they have vascular EDS. The answer also is no, that does not happen. These genetic changes are highly stable. If there is truly more than one connective tissue diagnosis in the same family, it is very likely just a rare coincidence. It doesn't mean that a gene change is doing different things in different people. How about most but not all of my findings can be explained by my genetically confirmed diagnosis of vascular EDS? I must have a new and rare disorder. No, that would often generally not be the case, but people with vascular EDS are at the same risk for all of the other common and rare things that affect all the rest of us. So if someone with vascular EDS happens to have a severe eye problem or early atherosclerosis, it's likely that they have that on the basis of another cause, that it's not part of their EDS diagnosis. All right, so we know that our RNA is translated and makes a protein. And the first thing we're gonna consider is how that can go wrong. So if you imagine a Morse code operator, and they're trying to interpret the Morse code ... dash, 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 ... The key to their success is to know that it takes 3 symbols to encode 1 letter. So ... is an S, ... is an O, and ... is another S. The same is true for our genetic code. It takes 3 letters of DNA to encode a building block of protein. So we can see in the upper left box here that if you have 3 T's in a row that the ribosome interprets, it would say that it's supposed to insert the amino acid or protein building block that's called phenylalanine, abbreviated as PHE. And you can see that every protein building block, every protein subunit requires 3 letters of code to tell the ribosome that it's time to insert that building block. So let's imagine that the ribosome is reading down our collagen RNA, and it has just finished reading the code CCA, and it knows to insert the protein building block proline at that position. The ribosome then moves along to the next 3-letter code, and it sees a GGA. It knows to insert glycine, another protein building block at that position, and then moves along correspondingly. You can see that that will end up with a full-length type 3 collagen protein shown as the green rectangle, and you can see the appropriate amino acid glycine was inserted at that first position that we considered. Now let's say somebody has a single DNA building block mutation. The G in the central position of the code has been changed to a C. Well, the ribosome doesn't know what's supposed to be there, so it's just going to read GCA as the code, and that will tell it to insert the protein building block alanine as opposed to glycine. So that is a mistake. That is a DNA change, and that can have consequence. You might ask me, well, why did I pick a glycine as our first example? And the answer is that collagen molecules are very picky. They require the tiny amino acid glycine at every 3rd position to allow the protein to fold properly. So you can see that 3 collagen molecules are coming together, and then they start to intertwine to form this so-called triple helical structure. That structure, that winding structure of collagen, is essential for its proper function. And as I mentioned, a glycine is essential at every 3rd position. So if you ask me what's the most common type of mutation causing vascular Ehlers-Danlos syndrome, it would be a mutation that substitutes something else for glycine within the protein. Now, this is a little bit of a complicated story, but it's important. So let's say this individual has 2 copies of the type 3 collagen gene. One that has a segment shown in blue, which is the normal sequence. The other that has a segment shown in red, which is the abnormal copy of the gene. Both of those genes will give rise to protein, the blue protein being normal, the red protein being mutant. Now, because 3 of these molecules have to find each other and do this dance, intertwine with each other, just playing odds, just based upon chance, only 1/8 of the time will 3 normal proteins find each other and intertwine. And 7/8 of the time, an abnormal red molecule is going to find itself into this triple helical structure. So that means that 7/8 of the protein that someone with vascular Ehlers-Danlos syndrome produces will be abnormal. Let's take a different circumstance. Same story as last time. Ribosome reads along at a position where a glycine should be used and the proper code is present, so a glycine is used. Well, let's say now that the first letter of that code, that first G, is changed to a T, shown as a red symbol. So now the code is TGA. It turns out that TGA is one of the signals to tell the ribosome to stop. Its job is done, but it's not in the proper position here. It happened prematurely. So this is now called a PTC, or premature termination codon. You might think that that would cause the cell to make a short protein, the ribosome stopped too early. But something completely different happens. Instead of making an abnormal protein, that premature stop signal causes the RNA to be degraded. The cell gets rid of it. It knows it's wrong. And that's a process that's called nonsense-mediated decay, but the net result is that you don't make any protein from that copy of the gene. So we go back to our cartoon. We still have one normal copy shown with the blue DNA segment, one mutant copy shown with the red segment. Now the normal copy is making normal protein, but the mutant copy is not making any protein. All of the protein chains that can intertwine with each other are normal. So you have half the normal amount of type 3 collagen because the mutant copy isn't making any protein, but everything that is made is of normal quality. So you end up having half normal function of type 3 collagen. Now, if you think it through, in the situation where you're producing mutant protein, only 1/8 of the collagen is normal. There's very little good protein. And in general, this causes a more severe condition. And you might hear physicians or researchers referring to this as a dominant negative situation. In contrast, if your type 3 collagen mutation signals no production of protein from the abnormal copy, you're left with a higher level of good protein, about 50%. In general, this is associated with a more mild condition, and this is a situation that you might hear referred to as haploinsufficiency. So does this really pan out? So this is like a lot of what I'll be telling you about in this talk. This is excellent work that's been done over many decades by Peter Byers, Melanie Pepin, and the group at University of Washington. And they looked at people with vascular Ehlers-Danlos syndrome that had glycine substitutions that were making very little normal protein. And they also looked at people with vascular Ehlers-Danlos syndrome that had one copy of the gene that made no protein, so-called haploinsufficiency. This curve is called a survival curve. So on the bottom x-axis, you have age, and on the y-axis, you have survival. You can see at the beginning of ascertaining or looking at these people, there was 100% survival. You can see that the people with glycine substitutions started getting into trouble at around the age of 16 to 18 or so. And then every decade, more and more people with glycine substitutions were having serious events that led to their demise. Whereas the people with haploinsufficiency were later in age before they started showing problems at around, in this experience, around the age of 30. And the rate of serious events was slower from that point forward. So yes, that did turn out to be true. It's better to have half the amount of type 3 collagen that you should have, but everything is normal, versus only having about 1/8 of normal protein. Well, there are 4 ways to encode a glycine building block. We're going to just choose one of them, this GGT code. you can see that, that if you change the first letter to an A, you get a serine building block added instead of glycine. If you change it to a C, you get a different amino acid called arginine. A T, you get a different one once again called cysteine. You can see that if you change the second letter, you get other amino acids that are substituted for the intended glycine. And the question that Peter Byers and his colleagues asked was, does it matter? Does it matter that your glycine was changed to a serine versus your glycine is changed to an alanine? The answer was yes, but only to an extent. So again, we're looking at survival curves here. The green line shows what was observed if someone had a glycine to serine change. The red line is showing what happened if someone had a glycine to valine change. You can see on average, overall, it was best if your glycine was changed to a serine, and it was worse if your glycine was changed to a valine. But for any given individual, it's essentially impossible to predict. You know, this individual here got into trouble with a serine substitution at around the age of 30, whereas this person with a valine substitution didn't get into trouble until around the age of 70. So for any given person, you can't make any firm conclusions, but on average, it's a little bit better if you had a serine as opposed to a valine. All right, now, folding of the type 3 collagen chains is known to start at the end of the molecules and then work its way to the beginning of the molecules. So as a very general rule, a change at the end of the molecule has the greatest impact on folding. The molecules don't even start to fold, and therefore can be associated with a more severe condition, whereas mutations at the beginning of the molecule allow the chains to start the process of folding and therefore can be associated with a more mild condition. But once again, there are many exceptions to this rule. Now, we're going to turn our attention now to this splicing process where you join blocks of RNA together to form this mature messenger RNA. That's not a random process. It's very precise. The machinery that performs splicing looks for signals in the DNA that tells them where one block ends and where the next block begins. And as you can imagine, mistakes happen in those DNA sequences that interfere with the splicing process. So here, once again, we have our pre-mRNA, and under normal circumstances, it undergoes splicing to form a mature mRNA that has an intact triplet code, starting from the start codon and going all the way to the intended termination codon. Ribosome latches on, reads along, and makes a full-length normal protein. And that person does not have any medical problem on that basis. If someone has a splicing error that tells the splicing machinery to skip a block of coding sequence that is not an even multiple of 3 DNA building blocks, the ribosome is going to get confused. It latches on, but it's going to read into that sequence where a piece of DNA is missing that's not a multiple of 3 nucleotides. That invariably is going to lead to a premature termination codon. It throws off the code, everything downstream of that. But we know now that that's going to lead to decay of the mutant RNA and a mild form of vascular EDS. If, however, a splicing error tells the machinery to skip a zone of coding sequence that is an even multiple of 3 nucleotides, the proper frame for the code is going to be maintained. It's not going to get shifted. So the ribosome will latch on, it will read all the way through, it will make a stable RNA that will make an abnormal protein that's simply missing a central chunk. So it's going to make a protein that can interfere with the use of the normal protein, and that would cause typical to severe vascular EDS. So if somebody says you have a splicing abnormality, that's not enough information to predict whether that might be associated with a mild form of vascular EDS or a more severe form. Now, there are splicing errors that have been found all throughout the type 3 collagen gene, some at the beginning of the coding sequence, some at the end. The question that Peter Byers and colleagues again asked, does it matter whether it's at a so-called splice acceptor or a so-called splice donor? And the answer was yes, but again, only to a modest extent. So the red line is showing the performance of people that have a glycine substitution. The blue line, people that have a splicing mistake in the splice donor. The green line, a splicing mistake in the splice acceptor sequence. And, by and large, everybody did about the same. So I guess the bottom line would be that that level of detail is not important. The bottom line is that there's lots of ways to make a mutant protein, and all of them in general would be predicted to cause a somewhat more severe condition. There's also lots of ways that the cell could simply not make any abnormal type 3 collagen protein. And in general, once again, that would be predicted to be associated with a more mild condition. So I'm not going to go through all these rare stories, but just suffice it to say that there are a variety of ways to get to these outcomes. So one question that I hear a lot from people is, why bother testing? I know my mom or dad had vascular EDS or has vascular EDS, and you know what, I'm not sure it makes a difference, so why should I test? Well, we do believe that in many circumstances that it does provide valuable information to get back the answer of a confirmatory test. It tells us which people who are at risk require imaging surveillance, which people might require a medication or recommendations regarding what activities they can safely do and what others they should avoid. We do know that knowing the mutation can provide some information about prognosis. And importantly, we believe it's very important for someone to learn that they do or do not have vascular EDS as compared to the hypermobile form, or even as compared to Marfan syndrome or Loeys-Dietz syndrome, because the management principles are different for various connective tissue disorders. It lets at-risk family members know quickly, once you know the mutation for a given family, whether they need ongoing medical care, whether they need to take precautions, whether they should frankly worry or not. And then for many families who consider use of family planning methods, knowing a mutation can allow that to happen by prenatal or pre-implantation genetic diagnosis. In some circumstances, you might hear the story that you shouldn't go ahead with genetic testing because the results are too hard to interpret. That can be true for some conditions and for some genes. Thankfully, for the type 3 collagen gene, and again, through the hard work of Peter Byers and others, we have learned a lot of good rules regarding how to interpret a genetic test. So there are 5 possibilities of a result that you get back. Something is likely pathogenic or causative, varying all the way down to something is innocent. It's not causative at all. With this gene in this condition, we are pretty good, in fact, really good at interpreting a DNA test. So I would not be too concerned about getting back an ambiguous result. There are a few other genetic determinants of disease severity. You may have heard that males with vascular EDS on average seem to get, begin the process of having serious events at an earlier age. In general, this coincides with adolescence in males, whereas females don't start showing problems until a somewhat later age that in broad terms coincides with childbearing. Again, there are many exceptions to this rule, but it's something that you may hear about. What about those skipped generations that can occur in some families that we talked about earlier? So that is a phenomenon that we believe is due to so-called genetic modifiers. As I mentioned, in vascular EDS, people have an abnormality in one copy of their type 3 collagen gene, but they also have lots of variation in the other 20,000 genes in every cell. Those normal variants decide what eye color we have, what type of hair we have, how tall we are. And you know what? They can also help determine how severe our vascular EDS will be. If you get dealt a favorable set of modifier variants, you might be relatively protected from the effects of a type 3 collagen mutation. Whereas if your brother or sister were dealt a less favorable hand or complement of variants in other genes, they might have less protection and show more severe disease. At an extreme circumstance, someone could get such a good set of modifier variants that they might be completely protected from the consequence of their type 3 collagen mutation. That's the situation that we previously called non-penetrance. So I think this is a really exciting area of research. And in fact, my lab has really dedicated itself to understanding modifier variants in connective tissue disorders. Together with the labs of Bart Loeys and Anne DePape and Julie DeBacher, we identified these 5 truly exceptional families, not with vascular EDS. These were families with Marfan syndrome. They were very big families by and large. Everyone that's shown by a colored symbol, either black or blue, has the Marfan mutation. The people that are shown as black symbols had classic Marfan syndrome with severe vascular disease in childhood into early adulthood, whereas the people that are shown in blue had the same Marfan mutation, but had not yet shown any vascular problem at the age of 50 or even 60 or 70. So the blue shaded people are, in our imagination, got the good variants to protect them, whereas the black shaded people did not. And we decided to use genetic methods to try to find the modifier genes and the modifier variants, with the belief that if we could understand how the body and nature protects some people with Marfan syndrome, maybe we could mimic that protective effect with a medication. So using these families, we did go on to map the modifier gene in these families. It was on human chromosome 6. And we've even identified a single gene where we think the protective modification is occurring. We've created gene changes in this gene in mice with Marfan syndrome, and realized that changing this gene can protect mice from having the severe vascular consequences of the disease. We now have gone on to see dramatic genetic modification going on in our mouse models. Here I'm showing you two different lines of mice that we created with vascular Ehlers-Danlos syndrome. One line has an early glycine substitution toward the beginning of the gene. That mouse overall shows a more mild disease phenotype with an average survival of about 400 days. We made another vascular EDS mouse with a glycine substitution toward the end of the gene that we predicted would be more severe. And indeed, this mouse shows fatal vascular events with an average survival of only about 40 days. We've used this severe mouse model of vascular EDS for many of our studies. And fortunately, we've been able to find some medications that can protect even the severe mouse model of vascular EDS. That's only making 1/8 the normal protein and yet is having long-term survival without vascular events by blocking two cellular pathways that we call the PKC or ERK pathway. We've been able to observe that the mice with the severe vascular EDS mutation show early death due to vascular rupture shortly after delivery, something that we also observe in women with other connective tissue disorders, including vascular EDS. We've been able to show in those vascular EDS mice, even the severe strain, that simply avoiding breastfeeding is protecting those mice from death after delivery, and that we can achieve the same protection by blocking a hormone that's used to stimulate milk production in lactating women called oxytocin. We've also learned that male mice with vascular EDS do less well than female mice, that they also get into trouble or start getting into trouble around the time of puberty, and that if we use medications to block male hormones called androgens, we can prevent those early vascular events in the male mice with vascular Ehlers-Danlos syndrome. This is what I find most exciting, though. This is the genetic modification study that we've just completed. Here you can see that when we put our severe vascular EDS mutation on one mouse strain that we call the black 6 strain of mice, that they die early, and that essentially by 150 days of age, all of the mice have died from a vascular event. If we put that same mutation on a different mouse background that we call the 129 strain of mice, they're still normal mice, they just got a different complement of modifier genes, we see full protection going all the way out to a normal mouse lifespan of 2 years. And in fact, they never develop vascular events, despite the fact that they still only have 1/8 the normal complement of type 3 collagen. We've now bred the two strains together and made so-called mixed mice. And we've succeeded in mapping the modifier gene in mice. We know exactly what gene it is now, what pathway it affects. And we have learned that there are experimental drugs that can make a black 6 mouse look like a protected 129 mouse. These are brand new data that were done by an extremely talented student in my lab named Caitlin Bowen with the support of the Marfan Foundation. And we expect to be able to share that story in full detail very soon, hopefully within the next month or two. To end, there are some people that have been told that they have vascular Ehlers-Danlos syndrome, but the genetic testing comes back negative. Some of those people have a condition that's called Loeys-Dietz syndrome. Those people also have aneurysms and tears throughout their arterial tree, although most commonly at the aortic root, which is different from vascular Ehlers-Danlos syndrome. They also have easy bruising, problems in pregnancy, a risk of bowel rupture that is more rare than in vascular EDS. They have similar skin changes, including translucent skin with visible underlying veins. They can have joint hypermobility and frequently have club foot deformity, as is also seen in vascular EDS. But fortunately, we do know many genes that cause this condition. They're different from vascular EDS. They're not the type 3 collagen gene. So genetic testing can help us separate these folks out quickly. And that's important because their management differs from the management for vascular EDS. And as a final thought, there are many different forms of Ehlers-Danlos syndrome. The unifying thing in most, if not all, people with some form of Ehlers-Danlos syndrome is some degree of joint and skin abnormalities. But the important point is that each of these various forms of Ehlers-Danlos syndrome has a set of clinical features, a specific mode of inheritance, recurrence risks, different underlying disease genes. So there are good rules to use to diagnose these various forms. Fortunately, most of them don't have the vascular consequences of vascular EDS. Fortunately, vascular EDS does not have many of the serious consequences of some of these other forms of EDS, so it's really important to get a very specific diagnosis and to work with care providers that can help take the best care of you based upon that diagnosis. At the end, I'd like to acknowledge the people who contributed to the work that I shared from my lab. I'd like to once again acknowledge the important work of the Byers Group in teaching us all about the genetics of Ehlers-Danlos syndrome. I'd like to thank the many people and foundations who've contributed to this work, and I'd be happy to try to answer some questions. Thank you for your attention.
Thank you, Dr. Dietz, and I'm definitely excited to get to the Q&A. So I'm going to ask the first question, which is, can you explain the genetic reasons why VEDS family members who test negative have varying degrees of hypermobility?
Yeah, so if someone tests negative for a familial mutation that's been observed in another family member that's known to have vascular EDS, they don't have vascular EDS, but that doesn't mean they don't have some difference in the quality of their connective tissue just by coincidence. In fact, if you look at everybody overall, about 15% of people have something quirky about their connective tissue. Those were the kids in school that could bend their fingers backward, they could turn their eyelids inside out. You know, they got something different going on with their connective tissue, but it's not vascular EDS and will not have the same problems. It's just by coincidence that happens.
Interesting. And I had a question about the mosaicism myself.
Mm-hmm.
How is that? You said that you can look at that with looking closely at the genetic test results.
Yeah.
Is that a normal test?
So if you have a suspicion that mosaicism might be going on, let's say that you have 2 unaffected parents that have 2 affected children. Sometimes if you take a saliva sample or a blood sample or a skin sample from the parents and look very carefully, you can see just a hint of a signal for a genetic change that's present in some but not all of the cells in their body. Sometimes the genetic change will be present in blood but not saliva, and that lets you know that that person is mosaic. So it generally takes some degree of a suspicion for someone to look carefully enough at a genetic test to try to see a trace amount of signal for mutant DNA.
Okay, that makes sense. Thank you. So the next question. If someone has a genetically confirmed COL3A1 mutation and some of the clinical findings, but not all, or let's say even none, does that mean that they do have VEDS or only maybe?
So that's a good point. You know, how do we define vascular EDS? Is it based upon a genetic predisposition? Is it based upon a clinical presentation? You know, by and large, if somebody has the vascular consequence of vascular EDS and has a confirmed genetic change that we know is capable of causing vascular EDS, then that term would be used. That person would be labeled as having vascular EDS. If someone had a mutation that was known to be capable of causing vascular EDS, but didn't have a single physical feature of the condition, you'd probably get different opinions from different people as to what you should call that. That person should know that they are at risk for vascular EDS manifestations. Their doctor should know that they're at risk. But you know, again, whether or not you use the label would be a matter of opinion.
Okay. And we're getting more questions coming in. Just want to make sure, Dr. Dietz, are you okay with staying a little bit over time to answer more questions? Yep. Wonderful. All right, so I think the next question is about your research in clinical variability, which was the middle image on slide. I'm not sure which slide that was on, where the mutation of VEDS is mild. Would the affected parents and grandparents test positive for VEDS in the genetic testing? Oh, that was in the beginning.
I'm going to go back.
It's 2 questions in one.
So let's, so is it,
Maybe
yeah, I think I know this one.
This one, yep.
So these individuals, by the virtue of the fact that they're shaded gray in this figure, mean that they do have some features of vascular EDS. They would test positive for the mutation, and that would be present in every cell of their body. That mutation obviously is sufficient or capable of causing classic vascular EDS as seen as in the young woman at the bottom of the center panel. So all three of these people would be designated as having vascular EDS.
Thank you. And we've got a couple questions on your research. I'm going to try to combine them into one question because they're all very similar. Based on your lab's findings, what would be the next step in moving toward a human trial? Would it be a PRK inhibitor? Would it be the hydralazine and spironolactone? There's a lot of questions about oxytocin as well.
Yeah, so we feel strongly that good, large, careful clinical trials need to be done, that we shouldn't all just decide that we're going to assume that what worked in mice will work well for people. That doesn't mean that in certain circumstances, people would not choose to use an FDA-approved blood pressure medication like hydralazine for someone that they're seeing with vascular EDS who can't take other medications. It just means that that would not become standard practice. It would not become the standard of care without careful clinical trials. You know, my hope is that some of these medications are going to be moved forward to large definitive clinical trials for people with vascular EDS. There is a group of people forming a company that's called Rumpus Therapeutics that are intrigued about the possibility that a PKC inhibitor will prove to be the most safe and most effective way of caring for people with vascular EDS. So they're now doing important basic work and raising money to support a large clinical trial to test that hypothesis. I think that the modifier study is likely to move forward to testing in people fairly soon. You know, in the first step may be to find these families that have extreme variation in the severity of vascular EDS within the same family among known mutation carriers. If we could prove that the same gene modifier that we found in mice is also working in people based upon genetics, I think that that would jumpstart one strategy over another with regard to which one should be tested first in clinical trials. So that's the kind of work we're hoping to partner with others to do, is to look at the genetics in families with variability in the severity of vascular EDS.
Great, so it sounds like there's some exciting things coming.
There's lots of activity. There's more is going to happen in the next 5 years than happened in the last 25 years. I'm sure of that.
That's wonderful. I'm sure we're all excited about it. I know I am. The next question is about a variable of unknown significance. What is required to move a VUS, or a variable of unknown significance, finding to pathogenic? This person has a family history of aortic aneurysms, but they have a VUS.
So a variant of uncertain significance means that there simply isn't enough information to suggest that that DNA change is causing that person's medical problem. Now, I would want to know a lot more about how typical the features are in that individual for vascular EDS. If they had an ambiguous DNA change and also their condition wasn't typical for vascular EDS, let's say they had aortic root enlargement and no skin bruising and all sorts of other exceptions to the rule, you know, that would make it less likely. What you look for is whether other people are finding that same DNA change in people with known vascular EDS. That would move it up in significance to say it's more likely disease causing than not. You're just looking for a preponderance of evidence, and there are many different criteria that can be used. But the ones that are most significant are, is this change being seen in other people with vascular EDS? That would move it up. Is this change being seen in people with some other medical condition that's known not to be vascular EDS? That would move it sideways. It would basically say, yes, this is pathogenic, but your condition is not vascular EDS. And then if a variant started to be seen in the general population at a high frequency, people with no medical problem whatsoever, that would move it down. That would say, no, this is most likely going to be innocent.
And then it would be something else.
Yeah, and something else would be causing the medical issues that person is experiencing.
Thank you. The next person has many symptoms of severe hypermobile EDS and some symptoms of vascular EDS. She was diagnosed genetically as VEDS, but is it possible that she has two types of EDS or a strange variant of VEDS?
Yeah, the bottom line is that anything is possible, but some things are simply not likely. So if you just played the odds, it's highly unlikely that the same person has two really rare connective tissue disorders. That if it would require lightning striking twice. I think it's best to try to explain someone on the basis of a single rare diagnosis, realizing that there can be exceptions. But it's also important to realize that people with a single rare diagnosis like vascular EDS might just by chance have some other common condition that's causing overlap features. So now that we're doing broad DNA sequencing, something called whole exome sequencing, where we look at all 20,000 genes at once, we are finding interesting combinations of genetic variants. One variant causing a rare disease like vascular EDS, and then in that same person, a variant known to cause a common problem that involves the joints, and in that same person, another variant known to cause a different common problem known to involve the eyes. And at the end of the day, when you look down at all of these variants, you say, aha, of course, that's what it obviously was, but it wasn't obvious until you had all those answers in front of you. Is it likely that this person has two rare, serious connective tissue disorders going on at the same time? It's possible, but not likely.
And that would be possible, say, if both of their parents had a different rare genetic condition that they each passed on, right?
So that can happen. So a family history can help you to try to figure that out. Sometimes people have more than one new mutation, so you don't get any hint from the family history. It's a new change in that that individual. But what certainly is not going on are strange hybrid connective tissue disorders that are something brand new and different. Most often a person can be explained based upon the combination of rare and common DNA changes that are causing conditions that affect people in the general population.
Okay, thank you for explaining that. And
Sure.
we see a lot of that quote unquote overlap touch of I really appreciate that. If you have all of the symptoms of vascular EDS without the hypermobility, do you still have vascular EDS?
Yes, and in fact, joint hypermobility is not all that common or all that severe in overall in people with vascular EDS. Again, there will be exceptions, but most people with vascular EDS do not have the same severe degree of generalized joint hypermobility that's seen in people with hypermobile or classic EDS. And if people with vascular EDS have joint hypermobility, it tends to be most prominent in the distal extremities, for example, in the hands, rather than the hips and the elbows and the shoulders, for example. So yes, joint hypermobility is not an obligate finding in people with vascular EDS, and many people with that diagnosis don't have joint hypermobility.
Thank you. And I'm going to ask one more question, and then I'll have a couple of announcements at the end. Would vitamin or mineral deficiencies, especially vitamin C, play a part in causing COL3A1 mutations?
So will a vitamin or mineral deficiency cause a mutation to happen? The answer is definitively no. Can a vitamin deficiency aggravate or make more severe the consequences of a vascular EDS mutation? And I think the answer to that question is likely yes. So vitamin C, for example, is a very interesting player. There are many enzymes, many proteins in the body that require a normal amount of vitamin C to perform optimally. Among those proteins that are vitamin C dependent are many proteins involved in making collagen molecules and in making collagen molecules strong. So for example, there is an enzyme that creates little bridges between collagen molecules that we call crosslinks, and those bridges help to make the molecules strong. Vitamin C will not, by and large, allow you to make more good type 3 collagen, but it may help you to make better use of the type 3 collagen that you have. So that's why many people, including myself, recommend that people with vascular EDS take high doses of vitamin C. It will make those proteins work better and should increase the strength of your collagen molecules. And in fact, we just finished testing that in vascular EDS mice, and we did find that vitamin C supplementation helps. So I think there's now even more evidence to support that.
Wonderful. And I do actually have one more question. We've had a couple people ask if you would wear red this Friday for Reds for VEDS Day.
Absolutely.
Thank you. And if you haven't heard about Reds for VEDS Day, I will share information on that in a moment. Thank you, Dr. Dietz, for your time tonight. I very much appreciate it. And thank you, everybody, for participating. I am going to go back to my PowerPoint here. Can everybody see my screen?
Yes.
Awesome. Okay, so if you liked this webinar, stay tuned for more webinars specific to VEDS. We'll announce them as they are scheduled. And before we say goodbye, I'm just going to take a few minutes to tell you ways to get involved in research, the community, and the VEDS movement. If you haven't already heard about the VEDS Collaborative Natural History Study, this is the largest study in the United States for VEDS. It's currently enrolling participants and is led by Dr. Shireen Shalhoub at the University of Washington. You can enroll in the study by emailing the VEDS Collaborative or by indicating you wish to enroll through the Backpack Registry. If you haven't already heard about Backpack Health, it's an app for your smartphone. It has a couple important functions. One is to compile the health records for you and your family members. It is safe and secure, comes in 6 languages, and has functionality that allows you to share certain information with others in your circle as you wish, which could be the school nurse at your child's now virtual school, with family members, a new doctor, or even EMTs in an emergency situation. The other function is for research. It's open to everyone with VEDS, and after you consent into the registry, your information will be de-identified and aggregated with information from other people in the registry. This will make it possible in the future for researchers to answer questions that can impact your life, your children's lives, and future generations. Joining Backpack and the registry is free. To join, you can go to the VEDSmovement.org and visit our healthcare management tool page. There's a lot of information there, and our friends at Backpack are always available to help. You can also indicate that you wish to join the VEDS Collaborative Natural History Study if you are enrolled in the Backpack Registry. If you are looking for more ways to get involved with the VEDS movement, we are always looking for members of the community to lead Team VEDS at our upcoming, now virtual, Walks for Victory on June 6th. You can visit marfan.org/walk for more information about these, and be sure to name your team Team VEDS and let us know you're walking for VEDS. We're also excited to announce a worldwide virtual walk for VEDS on October 3rd, which comes around the anniversary of the VEDS movement. We are preparing more information and planning a VEDS Awareness Week leading up to this event, so please stay tuned for more. We're very excited to be doing this. If you are looking for other ways to connect with the VEDS community, this Friday, May 15th, is the annual Global Reds for VEDS Day. Be sure to wear red, take a picture, and share the hashtag to raise awareness for VEDS with the global community. And of course, we will be getting a picture of Dr. Dietz wearing red as well. And to connect with others, we have 5 monthly virtual support groups, as well as a new friendly connection program to facilitate one-on-one connections between individuals. You can visit the VEDSmovement.org to learn more. And if you have a question, remember you can always contact our Help and Resource Center. We are here for you. This is the contact information for Jan Lynch, our wonderful nurse who leads the Help Center. Thank you, everybody, and have a wonderful night.
Thank you, Katie.
Thank you.